up in the morning these days. So hopefully some more folks will come in as we go forward. So today we're going to finish up the sets of lecture questions, and I did move their due dates. So the due dates now on assignments should be correct if you pay attention to what they are. What I'd like to do is today, besides finishing up the lecture questions, is have some review. If it doesn't have enough time in the lecture to do that, then we can use some of the lab time, because at this point, the lab that I'm assigning you between now and next Tuesday would be starting the digestive system. That was the original plan, if I remember rightly. Oh, no, not today. That's next time. Today, we're going to do the acid-base Physio-X. So that one shouldn't take too long. And so we should be able to go through the Physio-X answers for the urinary activities and any questions you have on that. And both of those are actually good review more for the exam than for the lab practical. Not to say that I wouldn't ask anything about them on the lab practical, but they are kind of put here more to get you ready for the upcoming exam so that you can play with variables that affect glomerular filtration rate, what hormones are important, and acid base and homeostasis. And so that's the plan. Again, the exam, and if you somehow missed the announcements, was moved till after the holiday, which means, and this is where my brain was a minute ago, which means next Tuesday, we can start digestive system. And I'll probably take some of the lecture time to make sure that you're ready for the exam, check in with you about that. The first part of digestive system is quite a bit of anatomy. And so it may be good to kind of go over the things that you'll be focusing on when you get into the lab. So that day, next Tuesday, you're starting the lab drawings and the models for the digestive system. So I have uploaded those into the study guide now. I'm trying to help you since there's a lot of testing going on. I'm trying to help you by putting more into the study guide. More guidance of what's on the slides or the models than maybe occurred in the first practical, more like the reference slides I put in for the second practical. But there is a lot of anatomy for the last practical, so you'll see there are a lot of slides. So make sure if you use this latest version of the study guide. Now, the thing that's missing from the study guide still is the digestive chemistry. I may fall back on using Physio-X again for that. I used it last semester in spring. It wasn't ideal. I have asked Kevin if he can make a video, and I haven't heard back about that from him. So there could be a handout that will appear, and that's our last lab of the semester, so we have a little time yet to worry about that. But what I'd like to do now that more people have come in is to set you into your groups and start you working on the acid-base homeostasis. I do think that this part is a little bit challenging, and that it also help you with the last part of the class, because we'll be talking about type A cells in particular, where the type A cells and the cells of the stomach I mentioned last time, the parietal cells, behave quite similarly and use similar transport mechanisms and have similar outcomes in what happens to the movement of bicarbonate and H plus ions, but for entirely different reasons. In the case of the kidney and the type A cells, they're going to help us secrete H plus ions, which is the only way our body can do that. For the stomach, they're trying to secrete the H plus ions in order to help you denature your proteins in your stomach and kill pathogens. So this is going to have kind of a double benefit if you get this down. Before we get there, we did touch on, and we'll come back to, some of the role of your respiratory tract in helping you maintain a normal pH. So this is your fastest way of responding to changes in your blood plasma pH, is to use your respiratory tract and carbonic anhydrase in the red blood cells to try and move that balanced equation, if it's too acid, to the towards the left, so CO2 is expelled more rapidly, and if you're alkalosis, to stop breathing for a while, to try and build up H plus ions and quickly maintain your acid-base balance. But in the long run, if your respiratory tract may be limited in how much it can do, because it's actually not really expelling H pluses, it's just converting them into water using carbonic anhydrase. All right, so with that early preamble, it looks like there's something in chat. Let's see. See that the study guide's been updated. Are there histology slides uploaded, or is that in progress? Maybe I haven't uploaded them. I will make sure that happens. I see them in my folder, but maybe I just put them in the slides in the PowerPoint slides of the study guide without having separate. So thanks for reminding me about doing that. All right, so you don't need them today, but you are going to need access to PhysioX. So let me start by getting breakout rooms set up. And we're going to make 5 of them again. And I will rename them with the questions and the letters. So (19) questions, 20, 21. Oops, that should be renamed, that one. Should be C. Okay, and this will be D. Questions 22, 3, and E. Okay. All right, let's see. I've got about 20 people. So that's 4 people in a room, except this first. Let me just see. All right, so I'll make that first group a little smaller. 1, 2, 3, okay. And assign, and then B has 3 questions. Yeah, 3 has 3 questions, so I'll make 1, 2, 3, 4 people in that one. And that makes this one, we'll have 1, 2, 3, 4, and 9. 1, 2, we have one more. Oops, only got 3. Well, you may end up, we'll see, because people do show up a little later, hopefully. Since it won't let me change the time once you guys start going into the rooms. I don't know why. That seems like a thing that would be important to do, but so I had to manually shut you out. I hope I didn't get you mid answers. So let's start with Group A, and let me see where in these questions. So Group A is plunging right into. Maybe before we start with Group A, I want to back up for a second and share a slide with you that some of you may have missed because it's really, I think, only embedded in the lecture slides. And it's something I put together about chemical buffering. And the lecture questions kind of jump over chemical buffering a bit. So let me just make sure we share this slide, and then we'll get into the renal system in a second with the lecture questions. Let me move this around. Get rid of this for a minute. Where can I put you guys? All right, so I put this together a while back because they do discuss all of this in the book, but there's some things that are kind of at a further away in the book after they do the renal system. So in intracellular fluids, if you remember in our electrolyte discussion, phosphate is relatively high in intracellular fluids and very low in extracellular fluids in general, low in extracellular fluids. And therefore, phosphate can act actually as a buffer, and it can accept H+ ions or release them when you're in the intercellular fluids, but it doesn't really do that extracellularly. Now, the exception to that, and we will get to there, I think it's one of the lecture questions, is urine. So we'll talk about that a bit later. In the intercellular fluids, proteins are the other main way in which H+ can be absorbed. So wherever you're having a hydrogen bonding occur, and a lot happens in proteins, then a certain number of H+ ions can disrupt that hydrogen bond without denaturing the protein to the point of it not being functional anymore. So within the cell, as it generates acidic waste, and one of you guys is going to talk about where all these acidic wastes and what they are being generated, then a certain amount of this buffering can happen in the intercellular fluids, but the cell is going to be pumping out H+ ions or lactic acid, acidic products that we'll talk about more later, into the extracellular fluids, because the pH in intracellular fluids should remain around 7, not 7.35, 7.45 like the blood, but rather it should be neutral at 7.0. So in order to do that, it has to expel these acidic waste products that one of the groups is going to be talking about into the interstitial fluid, and then that is picked up readily into the plasma. And there in the plasma, you also have a lot of proteins. If you recall, albumin is the main one. It's 60% of the protein, but there are the others, the globular proteins, and even the cells have proteins. Hemoglobin, it picks up a lot of those H+ ions. You know that because it helps using the Bohr effect to unload the oxygen from the hemoglobin. So there's chemical buffering going on, but you continually are making these H+ or acidic waste products that then end up in your extracellular fluids. So we are going to remind you from last time that then your respiratory system is going to be your fastest way to try and keep the pH of extracellular fluids of your blood within normal range here. So within the blood, bicarbonate they call a buffer. In a way it is, in that it can accept the H+, but when it does, it's most efficiently doing this with the help of carbonic anhydrase. So this goes back to what we did in the respiratory system, where the red blood cells do rapid conversion of bicarbonate H+ into CO2 and water. So our respiratory mechanisms will help bind up those H+ into water, but it's still not getting rid of them. And so finally, the urinary system, which is where we're going now, is going to be the slowest way, because after all, you don't make urine as quickly as you can use your respiratory system. It's going to take maybe hours to generate enough urine that you can, as it goes by, you can expel the H+ into. So the other problem that the kidney has is it's going to have to make sure that there's sufficient amount of bicarbonate available so that your respiratory system can adequately function to combine that bicarbonate with H+ and breathe it out as CO2. So in itself, bicarbonate is not necessarily a buffer in your blood, but it is a buffer when you combine it with the carbonic anhydrase in the lungs to breathe it out as CO2. I don't think that is well portrayed in the book. It's kind of like piecemeal. You have your chemical, you have your respiratory, and then you have your renal mechanisms. And I'm trying to sew it a little bit better together, partly using this figure, the idea that intracellular fluids first have to buffer things chemically because that's their only choice, but they're going to be expelling CO2, which will generate some H+ in the extracellular fluids just spontaneously, but also they'll be expelling acidic waste that we'll be talking about. And they have to pump out these H+ ions or acidic waste products because they're trying to maintain a neutral pH. Then we're going to turn to respiratory mechanisms to use the buffering of the blood to eventually convert those H+ into CO2 and water and breathe it out, but there's a limit to that as well. And so your renal systems will then be the only way you can actually expel CO2, but you also, expel H+ ions, but you also need to make sure that bicarbonate levels stay normal so that you can use your respiratory system. All right, I just repeated myself, or if there are any questions on that, you can just put that into chat and we'll go forward with our group A. So let me stop sharing this and I'll start sharing my notepad so that if you want, you can take some notes on this. Okay, so who is our spokesperson for Group A? I have only on this list here, Alex and Augustine.
Questions? I didn't look at what group I was in. I only looked at what questions I was doing.
Yeah, Dr. K, I think I was in that group too, but it looks like you assigned us 16 and 17. We weren't group A.
Oh God, I did it again and used the numbers from my old edition. Okay, so nobody did 14 and 15, is that? Which one did you do, Jeanette?
We did 15, we did 16 and 17.
Yeah, I don't know how I did that. I changed the numbers on it by writing over the top, but I still gave you the old ones. Okay, so you're starting with the actions of carbonic anhydrase in the membrane to reabsorb it. All right, let's just briefly, because they are really short ones, go through 14 and 15. So it says, describe the parts of the nephron that conserve, i.e. reabsorb. So I don't know why they changed the name from reabsorbing to conserving, but they did. So when the glomerulus filters out things, it's going to filter out bicarbonate, which you want to recover. You have to bring it back into the blood to keep those levels high enough. So whenever they're saying conserve, what they mean is the actual reabsorption by the PCT of bicarbonate. But the PCT and the can also make what's called new bicarbonate. Now, in the case of the PCT, it is truly new in that it was never carbon dioxide or water. It's being made from an amino acid, as they'll describe shortly, glutamate. And in the collecting duct, we'll see also there'll be some new made as well. So what describes the parts that conserve or reabsorb? That would be the PCT, which reabsorbs most everything. And which parts generate new? Well, part of the PCT, the very last part before it becomes the descending loop, can make new bicarbonate out of glutamate. And then the type A cells make new bicarbonate. In this case, they're making it out of CO2. So someone could argue, is that truly new? Because CO2 goes rapidly back and forth between being CO2 and bicarbonate when there's carbonic anhydrase around. But regardless. All right, so that's 14. 15, why is it necessary for H plus to be secreted? Well, I kind of answered that a few minutes ago, because your cells, almost all their metabolic wastes are acidic in some fashion. So they could be an acid themselves, like lactic acid being released, or they could be acid waste in the cell that then H pluses are being pumped out of, or it could even be CO2 being generated and that being converted into H plus by combining with water. So, all right, let's go jumping into 16, which might move us even a little faster now that I've done 14. So who is the spokesperson for doing the question about starting with the actions of carbonic anhydrase in the apical membrane? Explain how bicarbonate is reabsorbed. Is that you, Jeanette?
Yeah. So we basically, in this diagram, we just said that the reabsorption of filtered bicarbonate is coupled to the proton secretion. So within your cells, carbon dioxide and water will combine within this cell to form carbonic acid. That's carbonic acid, right? Yeah. So CO2 and water will combine to form carbonic acid via carbonic anhydrase. And for each proton that's secreted, a bicarbonate will enter the peritubular capillary. Then your secreted proton will combine with bicarbonate in the filtrate, forming carbonic acid. So the cycle kind of repeats. And within the filtrate, the carbonic acid is broken down to water and CO2. And the cycle repeats itself when CO2 enters the cell, and CO2 and water will combine again and form carbonic acid via carbonic anhydrase.
Okay, so what are the ways, and is this part of this? Yeah. What are the ways in which that bicarbonate makes its way across the basolateral membrane here? It's shown here.
So it can either use the antiport with chlorine, or it will use the symport with sodium.
Right. So notice the key down here, that there's these dashed lines are meaning that this is a secondary active transporter, and you only have these in the basolateral membrane. There aren't any over here. So the apical membrane is totally impermeable to bicarbonate. So that means that you don't have any direct way of picking that bicarbonate up that's out here in the filtrate that got pushed out in the glomerulus. So you have to kind of cheat the system here by turning the bicarbonate out there into carbon dioxide, like she said. Since carbon dioxide can cross by simple diffusion, carbon dioxide will then enter going down its concentration gradient. Why is the amount of CO2 low in the cells? And why would it come both from the blood and the filtrate? Because you have carbonic anhydrase that's working constantly to convert whatever CO2 is in the cell into bicarbonate and H+ ions. So then you might argue, okay, we're secreting H+ ions here, which we are, but just like Jeanette said, the H+ ions that cross very rapidly get picked up by a carbonic anhydrase that is in the membrane. I don't know why they made it look like it's in the filtrate, but it's in the membrane, the apical membrane, and that converts that H+ along with bicarbonate into water and CO2 again. So this keeps cycling until you don't have hardly any, if any, bicarbonate left in the filtrate. So on your table, back when we looked at reabsorption mechanisms in the PCT, I forget the number on the table, but I could probably find it. One of the things it tells you is that about 85% of bicarbonate is reabsorbed in the PCT. But really, virtually all of it is, because by the time you get to the end of the PCT, even though these transporters are there, there's not enough to pick up the rest of it easily before the filtrate moves along. So that brings our bicarbonate inside the cell as CO2. We convert it back to bicarbonate inside the cytoplasm so that levels of bicarbonate start to rise to high levels in the cytoplasm. So this is a little like when we, got the glucose, remember, in through a sodium symporter. You have high levels in here. Then it was pretty easy to get it out by facilitated diffusion if it was glucose. But in the case of an ion like this, it isn't brought out by facilitated diffusion. It has two ways, just like she mentioned, the chloride shift, which they're showing chloride come in exchange for bicarbonate go out. This is considered secondary active because you use ATP to make all this high level of bicarbonate inside the cell. So if ATP is used, it's in this case making a gradient where bicarbonate is really high in the cell. And then it's keeping an electrical balance by each bicarbonate going out. It brings in one negative chloride. For whatever reasons, the way God made it, I don't know, there's a second way, and that is a symporter with sodium. So clearly sodium is going against its gradient, because remember, this pump is just going like mad in PCT cells. So sodium levels are very high out here in the interstitial fluid, but there's enough bicarbonate to drag a sodium with it into the interstitial fluid. So there's two ways here. That bicarbonate is just picked up passively along a concentration gradient into the blood. Okay, so is this hormonally regulated? No. And there's no hormone that makes us go faster. Remember, our goal when we are reabsorbing, i.e. conserving, is to get the bicarbonate out of the filtrate. Don't lose it in your urine, get it back to the blood. However, this is not hormonally regulated, but when we start looking at abnormal conditions, like when somebody has, say, a respiratory acidosis, then CO2 levels in the blood are going to rise. And if you supply more substrate to this enzyme, it will generate more product more rapidly. So CO2 rising in the blood would cause higher levels in the filtrate as well, which would feed the system and enable bicarbonate to be rising in the blood by the use of this carbonic anhydrase. Does that make sense? Okay, so you may have to study this figure because it's a busy figure. Sometimes what I've done in the past with this lecture is to, start with each step at a time and just add a new piece to the figure. So the only thing, in a way, cluttering up this figure is the sodium-potassium pump, because we're not really using it on the basolateral membrane. Where this becomes important, and I jumped over it, is how do you get the H plus ions out? Well, once again, there are two ways. You can do it as many things move, by allowing sodium to come down its gradient. It's a secondary active antiporter with H plus. Or there's actually just a proton pump that is a primary one. Either way, why there's two, again, I don't know why there's two, but there's two ways it can go out. You can start in here. I don't know why, but for me, it's much more logical to start out here, but it's up to you. You can follow along in these steps up here. But I almost find it easier if you make each, make it's almost like a cartoon strip. This happens first, and then you draw the enzyme and what happens with it, and then the next step, rather than this cluttered figure. But it's up to you. Okay, good job. One thing's just out of interest is that one way some diuretics work, diuretics make you, pee more, is to slow this carbonic anhydrase. And this carbonic anhydrase, and that ultimately means that less sodium comes in, because if you don't supply more H pluses to feed the sodium, then the sodium won't come in as much, and therefore less water will come back. I mean, clearly sodium is being transported in multiple ways, so it's not going to stop sodium. It just slows it down a little bit. Okay, that's just for fun. I wouldn't test you on that. Okay, so let's go to the distal one. Are you still the spokesman?
Yeah, I'll be doing 17 as well. So in 17, it's asking about the distal portion of the proximal convoluted tubule and how it can generate new bicarbonate and under what conditions. So when you have acidosis, your arterial blood pH will fall below 7.35. And so that proximal convoluted tubule will generate new bicarbonate from glutamine. And so within their cell, you have your glutamine enters via simple diffusion. And when deamination occurs and oxidation, you do have it breaking down into ammonium and bicarbonate. So then bicarbonate leaves into the peritubular capillary, while the ammonium will enter into the filtrate and be excreted out into the urine. So this is, how we would get more bicarbonate into our capillaries.
Okay, so the only correction, and I can see why you'd make this assumption, if we go back to our key here, simple diffusion is a blue arrow. They're not actually telling you how glutamine gets in. A glutamine being too big, it couldn't go by simple diffusion. So they just put an arrow in there. The other thing that I'd a little bit argue with them is that there would be any glutamate in the filtrate by the time you get to the distal end of the PCT, unless you have so much glutamine in your diet that you exceeded Tmax. But it's irrelevant. As long as you get some glutamine, glutamine would then be in the blood. So what she was portraying is that this series of enzymes only becomes active if your pH drops below normal. So it's great she started with that. These enzymes are induced to become activated when pH of the extracellular fluids is low. Otherwise, they aren't activated, and glutamine would just stay in the blood and be reabsorbed, and that would be the end of it. Apparently, the PCT down here at the end is no longer able to get any bicarbonate that's left out here, but I'm not totally sure of that. So don't worry about these terms. I don't typically test you on these. We actually do return to them a little bit in the digestive tract. When you take glutamine and you strip off the amino group, that's the deamination here, and it makes it into ammonia. Now, ammonia is a pretty reactive molecule, and so it's kind of toxic to cells. So you don't really want a lot of ammonia out in your urine. That would only happen if your pH got to be kind of low. So again, these cells are only kicking this in under low pH. And then I don't know if there's also a chloride shift transporter in here. They don't show it, so I wouldn't test you on it. So the way it gets out is just like it did over here, as being a secondary active transport with the sodium, this guy right here. And you'd say, why secondary? Well, again, these enzymes use ATP in order to do their work. So that makes high levels of bicarbonate in here, which then can drag the sodium against its concentration gradient. And that makes high levels of ammonia, which you're not dragging the sodium. Sodium's actually helping by coming down its concentration gradient, but that's still a secondary active transport mechanism. So with these cells and the type A that we'll talk about, know the transporters in which membrane they're on and what mode of transportation, secondary, primary, be able to discuss that. All right, so by the time they filtrate now, assuming your pH is normal, has left the PCT, you would have absorbed nearly all the bicarbonate. And you might say, oh, H plus are secreted, but most of those are turned into CO2 and water, and therefore you're not really secreting H plus per se there. The only real true secretion of H plus takes place later in the type A cells. So who is discussing 18 and 19?
I'll be discussing those.
Okay, is Jessica?
Yeah.
Okay.
So 18, so type A intercalated cells generate new bicarbonate from carbon dioxide and water, and then secretes hydrogen ions via the hydrogen potassium antiporter, and they are triggered by low pH. Type B intercalated cells secrete bicarbonate during alkalosis, which is due to high levels of bicarbonate, and they are triggered by high pH.
That's right. Okay, great. So I'm not sure why in the book they don't put this. I said this in the lecture slides if you listen to them. So there's actually two ways to get across the luminal membrane here. And one of them is a proton pump. So it's just primary active transport pumping it out. The other way is a more inducible transporter. And just like she said, when your pH falls, you get more of these guys. Now, the positive thing is they're going to be another way to get H plus out. The negative is when they do, they bring potassium in, and that potassium raises your extracellular fluid, your potassium levels in your extracellular fluid. So the potassium goes all the way through here. They're not showing it go through here, but it's going to end up increasing your potassium levels. So going back to electrolyte balance, if you recall, if you have a high potassium, you may end up inducing more aldosterone if it gets high enough, because that's one of the stimulus for aldosterone release, which will help you secrete your aldosterone, secrete your potassium and bring that down. But there's a cascade of effects, because then your sodium levels may go up higher than you want them to. So what I'm trying to paint here is that when you have an acidosis event and you're your blood becomes acidotic, and many patients, for many reasons, do get acidosis. What that can lead to is hyperkalemia, kalemia, the potassium being high, and then that can lead to aldosterone going up, which to bring down your potassium, which can then lead to hypernatremia because you're bringing in more sodium than you like. So this is one reason I said this a little bit like a detective story, because there's a cascade of effects that can happen, all arising from a problem with, say, pH. So these are inducible, but generally your body's making these acidic wastes. So I'm not sure if this one is inducible at all. They don't talk about that, but it's probably chronically somewhat active with your pH being normal, since it's a way that your body can secrete these H pluses. So how does the bicarbonate get back? Well, they're only showing you the chloride shift here, because in the case of the type A cells, you're not reabsorbing other stuff. You don't tend to bring the sodium back. As far as I know, they don't bring up the role of the sodium-potassium pump at all in this type of cell. So there is no picture in the book for the type B cells. So I won't ask you who's on their basolateral and who's on their luminal membranes. We do know it secretes bicarbonate when you have alkalosis. And the best I've seen, looking it up, and maybe the easiest way to think about it, is if I could take this cell and flip it over, so that now the chloride shift antiporter here is on the apical membrane, and the H plus ATPase is on the basolateral membrane. So when you have alkalosis, you'll start to take CO2, convert it into bicarbonate, but secrete that into the filtrate, and the H pluses that are generated will be sent back to the blood to help you bring your pH back to normal. So I don't know whether there's one of these in a type B cell at all, perhaps not. And it was just relying on this primary active transporter. Alkalosis is a relatively rarer problem with patients. So you probably don't have as many adaptations to adjust your pH back down if it's too high. Okay, so that brings us to, I know you guys are doing a whole bunch of questions. 19, right? We're on question 19?
Yeah, so for 19, phosphate acts as a chemical buffer in the filtrate by binding hydrogen ions. And the hormone that affects the level of phosphate is PTH.
Great, perfect. So phosphate might be there all the time, a certain level of phosphate you're secreting in your urine, or just not reabsorbing. Not secreting, but not reabsorbing. But when you have PTH, then you secrete it. And if you go back to your electrolyte balance, remember PTH causes calcium to be reabsorbed, and it also causes phosphate to be secreted. So there's always some phosphate in your urine. And in fact, with the casts that are the unorganized casts, phosphate salts are not uncommon, even in normal folks. So this is just that same type A cell. Nothing different from the earlier picture. They just added this idea that you're going to be able to use phosphate to buffer urine. So if we go back to that original, maybe I'll even try and do that. Oops, there it is. Chemical buffering, there's urine is using a phosphate buffer. All right, moving on to 20, and I think I can stop sharing this now. Moving on to 20, who is the spokesperson for describing the metabolism of macromolecules?
I guess I'll do it.
Yeah.
Basically, we have the metabolism of macromolecules produces ketones and fatty acids from fats, phosphoric acid from proteins, and lactic acid from sugars.
Yeah, and CO2 overall, which can also combine with water and form H pluses. So some of these things we're going to return to in the digestive system, because we'll be talking about what happens to some of these waste products that are acidic by the liver. The liver can also help to deal with this, but the kidney is going to filter some of these guys out, but it's also going to deal with the H plus ions that they release, and you saw how that's going to happen. All right, so now 21 is kind of what I was covering earlier, but let's hear it again about the intracellular fluids buffering things to keep them at 7.
I was having trouble finding stuff on the ICF buffers. Basically, what I have is that cell proteins and HPO4 buffers the ICF, and then any excess is moved into the ECF, where especially the blood and the blood proteins and HCO3 buffer the pH.
That's perfect. Yeah. So that's kind of what I covered with that little handmade table. Intracellular fluids are buffered with phosphate, which is relatively high in the intercellular fluids, and the proteins. And the proteins, obviously, you don't want to denature them, so you don't want too many H pluses. So therefore, you've got to pump out these waste products like the fatty acids, the ketones, the lactic acid. You're going to release that into the extracellular fluids as well as H plus ions. Okay, so describe 3 ways in which respiratory acidosis might occur and give pH levels. That's 22. That's a new group, right? Do we have any spokesperson? All right, I'm also going to share the screen again, because what I have on it is that infamous formula or balanced equation. It's not a formula. So you can hopefully use that to your... Okay, so what I recommend is, and it's fine by me if you have a little scrap paper with nothing on it, and this in the real, when we meet in person, I certainly recommend you write this down from memory. So I'm going to trust your honesty that you'll write it down from memory before the test or somewhere, just to keep that in mind. All right, so 3 ways in which respiratory acidosis might occur and give blood pH, PCO2, and bicarbonate levels that would be signs of this condition. All right, so somebody has respiratory acidosis. What's happening? Who was the spokesperson? I forget now.
Kaylin.
Okay.
So the 3 ways in which respiratory acidosis might occur could be due to respiratory diseases that might cause impaired lung function or impaired ventilatory movement, or it could be caused by narcotic or barbiturate overdose or injury to the brain system, which may cause shallow breathing, which will lead to less CO2 being expelled. And if less CO2 is being expelled, that can lead to respiratory acidosis. And since less CO2 is being sent out than normal, that prevents the equation from shifting to the left, which will cause H plus and bicarbonate to build up in the blood. And since more H plus is being retained, that will cause the blood pH to drop below 7.35 due to the increased acidity in blood. So that would mean overall higher CO2 levels, low pH, and higher bicarbonate levels.
Great. So I kind of did them all in purple here. So why I say write this down is just like what she just said. Think about the lungs not being able to get rid of this CO2. That CO2 is going to build up in the body or in the blood. Just let's stay in the lungs here for a moment, and so that you're going to have higher levels of this when the blood leaves the lung. You're going to have higher levels of this when the blood leaves the lung. And don't forget that your body's still metabolizing. It's adding more H pluses. So you're unable to bind these H pluses up into water in your lungs, and therefore it builds up. And so eventually your blood pH drops below its normal levels. And that's going to then kick in compensation from your renal system. So one of the confounding or confusing things in the way the book, it has a nice little table on 1034 called A Closer Look, but it says with compensation, without compensation. So what we're going to assume, and I'm not sure if the book is always assuming this, but what we're going to assume is if I say with compensation, that means that the renal system has kicked in. If I say without compensation, that means we still have respiratory compensation, because in reality, your respiratory system is going to act so fast that you're not going to have to worry about whether it's kicked in or not. Now, in the case of a respiratory malfunction like this, it's going to be trying to kick in, but it's not kicking in. So the only thing you've got left now is your renal compensation. So in your renal compensation, it's looking at your H plus levels. It's not paying attention to that, right? So you may see it trying to secrete this and bring your pH down. And if you have respiratory acidosis, that's all you can do to try and bring your pH back, is to use your renal compensation. So remember, if it says with compensation, if I say with compensation, that means renal compensation. Okay. Don't worry about respiratory. We're just going to assume that always occurs. Okay, so that is great. Ventilation problems. Well, what diseases from your respiratory system could be ventilation problems like asthma, right? Or gas exchange problems, respiratory zone problems like pneumonia. And then the brain, we're talking about the respiratory centers in the medulla, may have, or the pons even may be malfunctioning, but they're going to then influence the medulla. Okay, so that brings us to 23, 3 ways you could have alkalosis. So I am going to erase all my purples. And are you doing that as well, the alkalosis?
No, that's going to be me, Professor.
Okay. Let me do some other color.
Do you want me to start?
Mm-hmm.
So the 3 ways that respiratory alkalosis can occur is due to strong emotions, hypoxemia, or brain tumors or injury. So this is when too much CO2 is expelled during hyperventilation. So if too much CO2 is sent out, this is going to push the equation to the left, and it's going to lead to a decrease in hydrogen ions and bicarbonate. So instead of being in the normal range of 7.35 to 7.45, the pH is going to be greater than 7.45.
That's going to increase, that's going to increase, and this is going to increase, right? And I forget, what did you say the 3rd way for the respiratory alkalosis? Panic attacks, strong emotion is what you said.
I said the strong emotion, hypoxemia, and brain tumors or injury.
Okay. So hypoxemia, can you define that?
No.
No. This is a condition of, I think that means, is that one on the list there?
Slide right now.
So one of you guys can,
Oh, okay.
but I think the condition of low respiration, I thought. Respiratory asthma, pneumonia. So it is low oxygenation. So this is low oxygenation. So that's probably where your respiratory system is kicking into having such low oxygen levels that you're compensating by breathing out your much more rapidly. For some reason, you have low oxygen, high CO2, and the reasons they give in the book are things like pneumonia and high altitude. So I'll put in here, because that's a complicated term, we'll put something like pneumonia, high altitude, where maybe high altitude you could relate to, because you might be breathing a lot more rapidly and deeper in order to bring enough oxygen in, since it's harder to get the oxygen to dissolve. All right, that was great. Okay, so again, we don't have any renal compensation involved in what we just did, right? If the renal compensation kicks in, it's going to start to aim at secreting the bicarbonate, raising the H pluses. So again, assume if I say with compensation, that I'm going to mean renal compensation is occurring. So you can assume if I don't say anything about compensation with renal, then you don't have to worry about how it's going to act. If I say, tell me now what the renal system has to do, then you have to worry about it. All right, the last couple questions are now metabolic ones, right? Okay, I'm going to do some erasing first, so hopefully I can use the same balanced equation here. Get rid of the green. Okay, so who is going to be in that last group and talking about metabolic? Oops,
that would be me, Noah.
Okay.
So with 24, we're talking about metabolic acidosis and the 5 ways that occurs. The first one that I have is severe diarrhea, and that's because there's a loss of nutrients before they can be resorbed. Renal disease, second one, is the failure of the kidneys to get rid of acids formed by the metabolic process. Untreated diabetes, because there's a lack of insulin, there's a lack of glucose being used up, which means fats are being used as a primary energy source, and ketoacidosis occurs. Starvation, which is a lack of dietary nutrients. Again, fats and proteins are then used, and this creates acidic products. And excess alcohol ingestion will create excess acids in the blood. And on the table, it says that bicarbonate levels, if this is inadequately compensated, bicarbonate levels are going to be less than normal. Is going to be less than 22 milliequivalents per liter, and pH obviously is going to be acidic. It's going to be less than 7.35. And I couldn't find anything really about the while it's inadequately compensated, talking about the PCO2 levels. But when it's being compensated, the book says during acidosis, it's going to blow off this CO2 to get rid of the blood of the excess acid. So the PCO2 will fall below 35.
All right. So what's happening here is what you're going on to say is, remember the tissues are adding H pluses by their acidic waste products here, right? And they're adding CO2, which combines with water, even just in the plasma, slowly, but it will make more H pluses. And the tissues are adding CO2. That's what I'm putting over here. So this is a metabolic problem. You're getting lots of this added in. Now this has the bicarbonate isn't being added in necessarily. I mean, if it's coming from CO2 being converted to H plus and bicarbonate, it is, but generally all this acidic, whoops, I put water, acidic waste, can't talk and write at the same time, acidic waste products are being secreted by your cells, and they're adding H pluses without adding bicarbonate here. So that's what you are alluding to, and that, the bicarbonate levels will fall in the blood. But the main way is that your respiratory system, this is a metabolic acidosis you're covering, right? Your respiratory system is going to try immediately to be compensating. So once again, we're going to say it's already compensating if it's respiratory. And you know that if your carotid aortic bodies detect an acidic level below 735, even if your PCO2 is normal, it will kick deeper and or rapid respiration, which will blow your CO2 off at a faster rate, even if CO2 is normal level. And that means your CO2 levels are going to, and this is what you said, right? CO2 levels are going to fall.
Right, they're going to fall.
And your bicarbonate levels are also going to fall because everything is going in this direction. I think the metabolic ones are a little harder than the respiratory problems, so you may take a little time to think about these. Are we okay? Any questions? I don't have chat open, but I guess it would pop up if there were. All right, so now let's do the other one where you have
Metabolic alkalosis.
Metabolic alkalosis. I'll try and make it yet another color if I find one. Okay, so the 4 ways. The first one would be vomiting or gastric suctioning. And this is because the loss of stomach acids. Selected diuretics would be a second one. Ingestion of excess sodium bicarbonate would be the third one. And the fourth one would be excess aldosterone. And ultimately, bicarbonate levels will be greater than 26 milliequivalents per liter, and pH will be more basic and greater than 7.45. Okay, I think I missed one in there, right? What was right before excess aldosterone?
Ingestion of excess sodium bicarbonate.
Yeah, so the kid that eats all the Tums or something, ingest.
And then to compensate this, the respiratory system is going to have slow and shallow breathing to allow carbon dioxide to accumulate in the blood.
Okay, so this comes to where we're going in the digestive system, and that's partly why I brought up the idea that the cells of the stomach, which make the acid for your stomach, are basically the same cell as a type A cell. They're working the same way. So your stomach regulates itself. It tries to keep itself at a very low pH of 1 to 2. And so if you start vomiting what's in your stomach, the fluids in your stomach, those cells are going to be triggered. They're going to say, whoa, the pH went up. I'm no longer 1 to 2. And they're going to start to be generating more acid. In generating more acid, they also generate more bicarbonate. And that bicarbonate ends up in your blood. So we'll come back to this in the digestive tract. So that's why excess vomiting or gastric suctioning, where some physician is, maybe you ate a poison or something and they're trying to take it out of your stomach, it causes the parietal cells of your stomach that make the acid to work overtime and make more bicarbonate, and you end up with alkalosis. Diuretics, we won't have to worry about why that is, but you saw I alluded to it affects the carbonic anhydrase is one way a diuretic might work. But the aldosterone, you can see if you have high levels of aldosterone, then potassium levels are going to be affected, but I wouldn't get too worried about that one. And again, the bicarbonate is going to end up causing a lower, push this equation over So if it pushes this equation over, if you suddenly add this from the outside, you're going to push this equation with your respiratory system over, which means you're going to end up losing more H plus than you intend to lose. Okay, I see we only have a little bit of time left. So with alkalosis, you saw that, again, not in your respiratory system, but say your digestive system, you may cause this to decrease, or you could cause this to increase by, say, eating a whole lot of Tums, and that could push everything towards that way. Okay, so the last thing I wanted to end on, and I did this out based on the that figure on 1034 that says a closer look. I like it, but I'm going to they don't give me a picture of that, so I can't share it with you. But I'd like you to kind of ignore or try and make sense if you want to, but it's a little challenging, of that little table at the bottom of it, because that's where it's using with compensation without defining what they mean by compensation. So I kind of redid it, and also putting this in context of what would happen in a medical setting. So say somebody comes in an emergency room and they take the person's blood pH and they find they have alkalosis. They don't know why there is alkalosis, but they see there's alkalosis. So they want to know, is this alkalosis from a respiratory origin or is it a metabolic origin? Because how you treat the person might be differently. So let's say it's a respiratory issue that the person's going to have. If you go back to that balance equation, which we can zoom back there again, if it's a respiratory problem, that means they're blowing the CO2 out at a high rate, and they might be doing that, this is alkalosis, because they're having a panic attack. Maybe I'm not sure if that's showing up for you. That means they're hyperventilating when they don't need to. They're not letting their normal regulation of CO2. It's being overridden by fear or some other strong emotion that's causing them to hyperventilate. So if you were dealing with a patient like that, you would want to know what their PCO2 is. If they're hyperventilating, their PCO2 is going to drop. And if you want to be sure, you could, which is harder to read quickly in an emergency room, but you could do a test to see what their bicarbonate levels are in their blood. These two are pretty easy to test rapidly. This is a little harder to test rapidly. So if you confirm this is a respiratory alkalosis, then you might, treat the patient by trying to, if it's a panic attack, trying to get them to calm down, or you might recall people are told to breathe into a paper sack, because then they'll rebreathe more CO2 back in, it's enriched CO2 air, and try and cause their pH to come back down to a normal level. So that's sort of the idea. I did it for all of these. If it's metabolic, if they have alkalosis and it's metabolic, then instead of seeing a drop in these two things, what you'll end up seeing is they have a rise in their PCO2, because their respiratory system, we can assume, is compensating. So if it's compensating, that person's going to have they don't actually breathe at a normal rate, they breathe at an irregular rate, which you'll learn in lab today. They'll skip breaths. So they're trying to do is, what their respiratory system is trying to do is push things back this way to raise the H plus levels. But when it does that, it's going to raise your bicarbonate levels and it's going to raise your CO2 levels. So that's this person. They have alkalosis. They came in, but instead of finding a low CO2, you're finding a high CO2 level, and you confirm it with a rising HCO3. Now notice I didn't put numbers on this because they're looking at this over time. Is it increasing or is it decreasing? Not necessarily a strict, their PCO2 is is 46 or something, you know. And in that case, it's a metabolic problem. So you don't want to be telling this person to breathe in a paper sack to try and cure their issue. So I did the same for acidosis. When we get into the lab in a few minutes, we can finish these two up. But I'm hoping what you can see by going back to this formula all the time and pushing things each way, if it's a respiratory problem, you're going to see a backlog of things and or an excess expelling of things that pushes the equation one way. If it's a metabolic problem, then you're inserting things on the right side of the equation, and then your respiratory system is not able to compensate enough for that, and you're seeing you're seeing it shift all to the left or to the right, depending on whether it's alkalosis or not. So we can finish this up. And the other thing I thought we could do as part of review for this, did I put it here? No, that's the wrong thing, is the problems at the end of the lecture set. So, and I thought I had them here, but I'm not seeing them. Maybe I hid it underneath this other window. Oh, here it is. Yeah, it's underneath this other window. What I'm talking about are these guys. So there's only 4 of them. Let me stop sharing that and start sharing this other one. Oops, dang. Too many quick clicks. Here it is. So at the very end of the lecture questions, lecture slides, I have one of these answered, I believe. So I give you a patient and give you the information from that, and then ask you to say, is this alkalosis? Is this acidosis? Is this high? And when I'm saying greater, this is really a rising or not, but I put them as numbers because I wanted you to also know they're normal numbers. And then I think the first one I do for you, but then there are a few more, which I hope I have in the ends of the slides. Yeah, I think I do, but my scrolling is not showing them. I have a few more of these that we could try as practice because I think it's important for you to practice. This is kind of like learning math. You've got to do these problems in order to know you can do them. All right, so that finishes up the last 2 essays. Essay 7, I believe, it's, if I have the number right, is no, essay 6 has some things about the respiratory compensation and metabolic compensation, which is a lot of what we just did here. And then 7 is on renal mechanisms, which we did at the beginning of this lecture. So I did post comments, the earlier comments for 1 through 4, and after today, I'll put the 6 and 7. And if you say, well, what about 5? Well, 5 is really done for you by that table. And then I sent you, my little arrow diagrams that are based on that table. So you have kind of a couple ways of studying those to help you study that. All right, I see one chat thing has shown up in here. Okay, so I will meet you back here at 11:10, and we can do some review, but also you'll be doing the PhysioX acid base, and that will be another way of helping to review. So I will see you all at just answering this person. Try. Okay, I'll see you all at 11:10. Okay. Okay, I'm going to stop sharing, and we can continue our both going over this and review there.